Welcome everybody. It's good to see you all in a sense, and thanks for attending Grand Rounds today. This is a special event from my perspective. First of all, it's our annual Peter Miller Musser Lectureship. It's also co-sponsored by Mass General Neuroscience and the Center for the Neuroscience of Psychedelics. The latter, some of you may be familiar with, others may not know that there is a center that's looking at how psychedelics change the brain and view this as an opportunity to transform the future psychotherapeutics. That this center has come into existence and has had some early successes in funding itself. It relates to a story, and today's Grand Round speaker, whether he knows it or not, is critical to that story because it was my attendance at a program. Actually, Franklin King, who I'll be introducing in a moment, was a panelist at that program, and our today's speaker, Robin Carhart Harris, was presenting, streaming a lecture on neuroimaging and psychedelics. And he, in a sense, rang a bell for us about the potential for using some of his insights and work and psychedelics to study a problem that some of us had been interested in. In this case, it was just rumination. But it led to being connected really to this remarkable world of people who are passionate about and interested in and researching psychedelics, and led us to start our own program here. So we're very much in Robin's debt for that moment that really triggered the center that we have today. So it's a pleasure to have him here, and I'm grateful for him to take the time. And I'm now going to turn it over to Franklin King. Franklin is one of the leaders of our effort. He directs the education and training component of the center. Others who are involved in leading the center are Bruce Rosen in the Martino Center for Neuroimaging, Steve Haggerty in the Chemical Neurobiology Lab in Neurology and Psychiatry, and Sharman Ghaznavi, who works with me as Associate Director and was also one of the early researchers on the Rumination Project. So this is an exciting moment for us, and Franklin, take it away and tell us about our remarkable today's Grand Round speaker.
Thanks so much, Jerry. And thank you everyone for attending. And it's my great pleasure to introduce to you Dr. Robin Carhart Harris. And I want to tell you a little bit about Dr. Carhart Harris before he gives us his talk today. So Dr. Carhart Harris moved to Imperial College London in 2008 after obtaining a PhD in psychopharmacology from the University of Bristol, and then an MA in psychoanalysis from Brunel University. At Imperial, Robin has designed human brain imaging studies with LSD, psilocybin, MDMA, and DMT, a clinical trial of psilocybin for treatment-resistant depression, an ongoing trial comparing psilocybin with escitalopram for major depressive disorder, and a multimodal imaging study in healthy volunteers receiving psilocybin for the first time. Robin founded the Center for Psychedelic Research at Imperial College in London in April 2019, and this was the first of its kind in the world. One reason for this is that a decades-long struggle to resurrect medical research in this area is beginning to bear fruit. So at Imperial, they have spearheaded work showing how psilocybin, which is the active ingredient in magic mushrooms, can be used to assist psychotherapy for treatment-resistant depression, making a significant difference when conventional antidepressants and talking therapy have not. Right now, they're crunching data from a much larger depression trial, comparing psilocybin-assisted therapy with a 6-week course of a conventional antidepressant drug, escitalopram, and I'm sure we'll hear about that today. Preliminary analyses indicate game-changing results. All this boils down to, I think it's safe to say that Dr. Carhart Harris is really the preeminent neuroscientist in the field of psychedelics today. And I'm looking forward to his talk. And again, we really appreciate your coming. And Dr. Carhart Harris, it's my great pleasure to introduce you. Take it away.
Thank you, Franklin. Well, it's an honor to be with you all today. When I was over in Boston, gosh, it was this year before it all, changed. and I was so warmly received, and so I regard MGH, Jerry, Bruce, and Franklin so highly, and really grateful and honored to be giving this talk to you today. Hopefully you can see my screen. I'll put it on screen share now. I'm really keen to get everything in, so forgive me if this is a little rapid. It's just that I want to cover everything and then give us a good bit of time for some questions at the end. So this will be a somewhat conceptual talk. There'll be some important principles I want to articulate, but it will also be, sprinkled with empiricism throughout. So let's start from the high end. What's the most precise way in which we can define classic psychedelic drugs? These are drugs like LSD, perhaps the prototypical psychedelic. It was only after it was discovered in 1943 that just over 10 years later, the term psychedelic was coined by the British psychiatrist Humphrey Osmond in conversation with Aldous Huxley. So LSD is an example, psilocybin is another, DMT is another, mescaline as well could be considered a classic psychedelic, albeit phenethylamine rather than a tryptamine. So if we focus on the pharmacology of these classic psychedelics, we come upon a definition in that all of these compounds stimulate a certain serotonin receptor, the Serotonin2A receptor. These receptors, one of at least 14 different serotonin receptor subtypes, which speaks to the particular complexity of this neuromodulatory system, the serotonin system. And this particular receptor, the 2A receptor, is very much a cortical receptor. You can see this agonist ligand PET image here. So we're labeling the functionally active Serotonin2A receptors here. And try and commit to memory this spatial pattern. We're going to see it again later on, but it's quite telling. You can see that particularly these transmodal cortical regions have a very dense expression of the 2A receptor. So the 2A receptor isn't just a cortical receptor. A lot of these serotonin receptors, you couldn't really say that about them. The 1A receptor has very dense expression in limbic regions, for example. You can see the hippocampus there in blue because there's relatively little 2A expression there. But these transmodal cortical regions, less so the unimodal cortical regions like the sensory motor strip. You can see quite light expression there, but these hierarchically high level, perhaps species specific, and we'll get onto that, cortical regions are where you find these key receptors that classic psychedelics work at. We know that this receptor is critically important because the affinity of a 2A agonist for the 2A receptor correlates very strongly with the potency of that drug. So LSD, very high affinity, very potent. You only need a tiny amount for it to have its signature psychedelic subjective effects. We also know that if you block the 2A receptor with an antagonist, so antagonist pre-treatment studies will effectively abolish any of the characteristic psychedelic effects of the subsequently given psychedelic. We also know quite recently now from PET imaging work that the occupancy of the 2A receptor by a given dose of, in this case, psilocybin, correlates very strongly with people's ratings of subjective intensity of the effects of the psilocybin. So you can see the percentage occupancy on the x-axis there. Once we get up around sort of 60%, you can see signs of, I know the sample size is small there, but you can see signs of an inflection where the intensity of the subjective effect starts to leap up. And around about the dose that we use therapeutically, around about 20 milligrams plus of psilocybin, is where we're getting this 60% occupancy and a quantitative and qualitative change in the subjective effects with the drug. So take home there, 2A receptor, serotonin 2A receptor, cortical receptor, high-level cortical receptor is the molecular gateway through which classic psychedelic drugs work, and how we can perhaps define them most precisely via their pharmacology. So what does the 2A receptor do? Well, we know very well that serotonin itself is involved in plasticity, and particularly the serotonin-2A receptor. We can look at markers of neuroplasticity, whether it's BDNF, which doubles in the cortex with administration of a psychedelic, in one particular case, DOI. We can look at synaptogenesis, as was done recently in this Cell paper, and marked increases in the growth of functional components of the synapse with a range of different psychedelics. We can look at brain development, the involvement of serotonin and the 2A receptor specifically there. Of course, a period of particularly high plasticity likely for the functional reason of learning. We can look at learning itself, low-level associative learning, learning rate being sped up with administration of a 2A agonist, in this case LSD, in a series of studies in rabbits, and administration of a 2A blocker, in this case ritanserin, slowing down the learning rate, low-level associative learning being retarded with a 2A blocker. We can look at extinction learning or unlearning, the kind of thing you might want to promote where a type of learning is maladaptive. You could think of addiction, for example. If you want to break certain associations, then you might want to increase extinction learning or unlearning, and that's also been shown to be promoted by administration of serotonin 2A agonist psychedelics. Cognitive flexibility or set shifting, promoted by 2A agonism and blunted by 2A blockade. Psychological flexibility, somewhat more abstract construct related to certain schools of psychotherapy, most notably acceptance and commitment therapy, has also been shown to be promoted subacutely after psychedelic administration. So why would the 2A receptor, in its association with the promotion of broadly defined plasticity, broadly defined as the ability to change, an increased ability to change, why might that be useful? Well, perhaps the most obvious extrapolation to make there would be that it would promote adaptability. Now let's forget about psychedelics for a moment and think about why this receptor may have evolved. It's most simple to assume that it's evolved for serotonin, the endogenous ligand. And so under what conditions can you upregulate this system? And this is really the product of a review that's just been accepted by the Journal of Psychopharmacology, written with a US student, Ari Brower, and myself. And here we review evidence showing that chronic stress in particular primes the 2A receptor system, whether it's increasing responsiveness to 2A agonists or increasing the availability of the receptor itself. And we review a range of different stressors. We show that physiological stressors like hypoxia, shortage of oxygen, actually markedly increases the availability of the serotonin 2A receptor. So we can think of chronic stress as priming the system, and acute stress seems to be perhaps the most reliable way of and robust way of releasing serotonin itself. So if you prime this particular aspect of the serotonin system, the 2A system, the one that psychedelics work through, and then you stress an animal, in this case you can see a chart in rodents showing serotonin release measured with microdialysis in the hippocampus, frontal cortex. You can see it doubling with tail pinch, and also marked increases with just handling. So these inductions of stress, acute stress, releasing serotonin. So we're arguing that the combination of these factors might be the natural conditions under which you activate the 2A system. So stress, adversity, what does that call for? Well, it arguably calls for an adaptive response, a plastic response. And that's what we're arguing is the sort of functional drive behind the evolution of this aspect of our brains. We also introduce this notion of the pivotal mental state and say that the psychedelic state is just one type of pivotal mental state. We could look at others such as trauma. We could look at psychotic states, particularly early psychotic states. We could look at psychological crises, perhaps conversion type experiences. What we do in this review is review a range of different studies that show similarities in the phenomenology between these states and, importantly, also their physiology and pharmacology. So that's the pivotal mental state. And the psychedelic state, we argue, is just one example, a drug-induced example. And the key message here is that these are pivotal states that can pivot depending on context. They're highly, especially, perhaps exceptionally dependent on context for where they pivot towards. They're hyperplastic states, and so exceptionally sensitive to environmental conditions, context more generally, and they can pivot perhaps towards wellness. You might think of a near-death experience or a conversion type experience, and then your life going along a more healthy direction. Perhaps you quit drinking and live a healthier lifestyle, or perhaps through trauma, you might pivot towards pathology. You might develop a chronic psychotic disorder. So this principle for me helps to articulate the importance, to express and highlight the importance of the context dependency of psychedelic therapy and the importance of the therapeutic component being brought into the hyperplastic foundational state, and then trying to move someone towards a healthier way of being. Reviewing now the latest clinical research on psychedelics, the best way to do this in the interest of time is to really focus on a few take-home messages. One is that the improvements in clinical outcomes, whether that's depressive symptoms, improvements in well-being, improvements in addiction symptoms, anxiety, these appear very quickly in now our second completed depression trial. We see improvements the next day. So it's very comparable to what's been promoted really with ketamine for depression. There's no sign that psilocybin is any slower in the onset of its therapeutic action. And actually there may be a particular edge over ketamine with these classic psychedelics in that the improvements often appear to be enduring. In fact, remarkably, these clinical studies have often involved just 1 or 2 dosing sessions with the psychedelic, and yet therapeutic outcomes are substantially improved in many of these studies, 6 months, sometimes 12 months, depending on the follow-up period, after just that 1 or 2 drug administration session. And so that rapid action, the enduring action, I'll in a moment talk more about the possible transdiagnostic action that a range of different disorders, a range of different symptom clusters seem to be sensitive to improvement with psychedelic therapy. If that's 3 take-home messages, rapid, enduring, transdiagnostic, then the 4th one would be context, that all of these studies, all of the evidence that is building for psychedelic therapy is coming from studies that have manipulated the context in a particular way. First of all, there's been careful screening and then psychological preparation of the participants. Then the participants are looked after, usually by 2 mental health professionals. There's music listening, there's eye shades, an aesthetically pleasing environment, there's aftercare, 1, 2, 3 sessions, sometimes longer in some of these other studies, the addiction studies with subsequent motivational therapy, for example, for tobacco addiction, Matt Johnson's work. So, there's a bookending of the drug session itself by psychological support, and that really shouldn't be glossed over. It's a very important principle speaking to the therapeutic model of psychedelic therapy. So a bit more on that potential transdiagnostic action and moving us towards coverage of some models of psychedelic action and psychedelic therapies, therapeutic action. And so we could look at a range of different symptom clusters, whether it's negative thinking, negative cognitive biases in depression, biases in chronic pain, the way that we look at ourselves, our bodies, our relationship with our pain, anxieties, whether they're phobic anxieties, attachments to particular objects of anxiety or objects of addiction, reward or relief in a range of different addictions. And you could characterize all of these, whether it's compulsions in OCD, as maladaptive habits. There's a term in evolutionary science, which is quite useful, I think, of canalization and Jerry's interest in rumination, which I also share. There's something related here that you could think of an energy landscape and a steepening of the sides that perhaps in certain conditions of uncertainty, anxious uncertainty, it might feel somewhat sort of self-serving, protective, perhaps a defense mechanism to channel oneself, one's behaviors, one's thoughts along a particular route, even if that sort of effort at adaptation is maladaptive and ultimately unhelpful. And so this is, for me, a useful model of thinking of not every psychopathology, but a lot of them, and perhaps those that are most sensitive to psychedelic therapy, or those that are the best candidates for psychedelic therapy. And what kind of treatment would you want to address this canalization, if you want, in thought and behavior? Well, perhaps an intervention that increases plasticity, increases adaptability, increases the ability to change simply. And so what I'm arguing is that psychedelics and psychedelic therapy, first of all, promote conditions of the relaxation of these habits of mind and or behavior. And under those conditions of relaxation, we can work with the psychological support to a healthier revision of these maladaptive strategies. Now, this is a bit of a switch, but I think it's necessary because a lot of my work has been focusing on the acute action, acute brain action of psychedelics. So these are brain imaging studies while people are under the influence of psychedelics. All of our studies that have looked in the brain under the influence of psychedelics have done so in healthy volunteers. So this is translational work, really for reasons of safety, although I do believe done in the right way, it can be done in patients, and that would address an important knowledge gap in the field. So these series of images, this one was particularly popular, looking at between system or between network communication being promoted under psilocybin. This work with LSD, a somewhat different analysis, seeding the primary visual cortex and then asking the question, where in the brain is activity fluctuating in synchrony with that seed region? And ordinarily, most of the coherent activity is confined to the visual system itself, but under LSD, much more of the brain is now fluctuating coherently, the spontaneous brain activity, eyes closed, resting state, the activities correlating now with V1. What does that mean? Well, one way we can address that is look at people's subjective ratings and then correlate the variance in those ratings, intersubject variance in those ratings with this brain effect. And when we do that, we see that there's a somewhat specific relationship between people's ratings of complex imagery. Remember, they have their eyes closed. Ordinarily, you don't see so much with your eyes closed, just sort of vague things, perhaps in your mind's eye, but under LSD, the imagery is very, very vivid, very abstract. And so the intensity of that effect is correlating with this brain effect. We've seen something similar with DMT, here looking at pairwise connectivity increases in red and yellow here under DMT relative to placebo, analyzed in a different way using an approach called functional connectivity density, otherwise referred to as global connectivity, functional connectivity. Now, these hot colors are somewhat misleading, and I'm going to tell you why in a moment, because you might see them and think, you know, they are regions where there's an increase in coupling or correlation in the activity with the rest of the brain, so an increase in global connectivity in those hot colors. But let's have a look at this dynamic analysis, because it starts to tell a more intuitive and I think intriguing story. Here, we're using this leading eigenvariate dynamic analysis developed by Joanna Cabral and Gustavo Deco. And here are a range of different states, globally coherent ones, slightly misleading that the brain, number one is sort of devoid of any color. Really, more accurately, it should be filled with color, because it's where all the activity over time is phase locked. It's all fluctuating in synchrony. So it's a globally coherent brain state. And these are a range of other states in which the brain can enter. There's no real meaning to the temporal sequence here. It's just on a chunk of data, say 10 minutes of DMT resting state fMRI data. We're asking at any given time, what state of this array of 8 are you most likely to be in? What network or brain state are you most likely to see in the brain at any given time? And when you look at placebo versus DMT, or pre-DMT versus post-DMT, you see that the globally coherent state, this first one in the sequence, is much more prominent under the DMT. And this is quite a well-replicated finding from our team, at least. We've seen this with LSD and psilocybin, now with DMT. So you go into this globally coherent state much more often under the DMT. And what states are you less likely to gravitate into under the DMT? Well, it's the default mode state, or default mode, and it's these frontoparietal states as well. So do commit that to memory, because that's going to be important. You know, back in 2010, I know people have said similar things about, you know, speculating about the default mode network. What is it really? And they've linked it to self-referencing, even the umbrella term of the self itself. And in 2010, I did a similar thing with a somewhat, I suppose, Freudian spin in collaboration with Carl Friston. We associated default mode functioning with ego functioning. And that's a hypothesis that I often keep in mind over time. It's felt less sort of default mode centric in my mind, and more about high-level frontoparietal networks more generally. Not just the default mode network, but these other high-level frontoparietal networks. And let's just remind ourselves, you're less likely, significantly less likely, to gravitate into these high-level frontoparietal states under DMT. Is this a possible neural correlate of so-called ego dissolution under psychedelics? I think that's a tantalizing possibility and needs to be looked at further as we go forward. We've done this kind of thing in the past, looking at default mode network integrity, a particular circuit within the default mode network, and the decrease in the integrity of the default mode network correlating with ratings of ego disintegration under LSD. All I would say these days is the truth is likely to be less specific to the default mode network. And also that question of specificity, you know, can we capture aspects of the phenomenology that relate to specific aspects of brain changes? I think is an ongoing quest. I think to some extent, yes, it's just there's a lot of overlap. You know, the more intense the ego disintegration, often actually the more intense the other aspects of the so-called psychedelic states. So it's really quite difficult to tease apart these different dimensions, if you want, of the the psychedelic state. But these other frontoparietal networks also break down under psychedelics and also correlate with ratings of ego dissolution or ego disintegration in our studies. Now, this is a slide I'm quite excited to show. It's fresh. I haven't presented it before, but it's one that is really exciting me in my reading at the moment. I get in trouble from the family on holiday when I'm reading around this work instead of, having a holiday or looking after the kids. But it's just so tantalizing. And let's go through it then. So first of all, we have the old familiar mapping of the 2A receptor, that the density is highest in these transmodal cortical regions. Now let's skip to this image. 2 and 3 are the references there. These are regions of expansion, evolutionary expansion from the macaque monkey up to the human, come upon through MRI work. Similar results are found when we look at chimp to human. It's just the profile there's more data really from macaque. So we can think of, the 2A receptor as being especially dense in these aspects of cortex that are so massively expanded in our species. There's something intriguing there. We can look at myelination, the weakest myelination being in these transmodal regions, stronger in the unimodal regions. We can do something like a PCA on the functional connectivity profile of the human brain and find that it creates this principal gradient where the unimodal cortices correlate with each other and are least correlated with the other end of the hierarchy, the high end, if you want, these transmodal cortical regions where connectivity strength is strong between those distally separated transmodal regions. In fact, they're the most distally separated from the unimodal cortical regions as could be possible, perhaps suggestive again of their special expansion as these cortical regions especially have expanded through evolution into humans. We can look at this amazing work from Jack Gallant and colleagues looking at semantic processing and in this case listening to narratives and then running a PCA on the atlas of where are regions sensitive to different words and phrases, and the more concrete words being associated with the low-level unimodal cortex, and the more abstract words being associated again with the transmodal context. We can look at connection length, that fibers are shorter, more local in the unimodal cortex and more distally connected in the transmodal cortex. We can look at the temporal hierarchy for movie watching and regions that have a slower response to components of the movies, and those being associated also with the transmodal cortex, whereas the unimodal cortex, at least here, the visual cortex, being more sensitive to short components, short aspects of the movie. We can look at between subject variability, and that being highest in the transmodal cortex, and we can come to a psychedelic and look at DMT and this increase in global connectivity with the same old transmodal cortical regions. We plotted this effect versus the 2A and found a relationship, as others have done, with things like gene expression as well, finding a relationship there where certain genes are disproportionately expressed in the transmodal cortex, so recapitulating these other anatomical maps. So there's something going on here. What is it? I'll refrain from anything grandiose about the 2A receptor, driving the evolution of the human brain. I'm not convinced of that idea. It was popularized by a certain Terence McKenna, I think in the 1980s, the so-called stoned ape hypothesis. I've never bought it. I still don't buy it. But I mean, the evidence is saying there's at least a tracking of cortical expansion into humans, and those regions that have undergone the largest expansion being those regions that express the 2A receptor most highly. Why is that? I don't know. When they're stimulated, there is a entropic effect on those regions. So it's not like they are, 2A stimulation is sort of driving the order or organization in that system, that network. If you want, rather the opposite. It breaks down. A number of our analyses and other teams' analyses of the integrity of this network, whether it's looking at the default mode or the frontoparietal networks, the integrity breaks down under psychedelics, as I said earlier, and that also relates to ratings of ego disintegration. So the effect is somewhat to scramble that system. So at least if you look at the acute state, there's not something obvious there that the functionality of that system is being driven in any kind of familiar, sort of canonical way by activation of the 2A receptor. Rather, it's being sort of, I don't want to say taken offline, although you could say that. It's more sort of being scrambled. There's this sort of entropic effect. I've used the analogy of a snow globe in the past, shaking the snow globe. So anyway, the entropic brain principle, what is it? It simply says that the entropy or the unpredictability of ongoing spontaneous brain activity, whether measured by EEG or MEG, we think we can do it with fMRI, and we can do it with fMRI, have done it with fMRI sample entropy. That tracks the richness of content, the informational richness of ongoing subjective experience. So, for example, if you are knocked out with an anesthetic, the entropy of brain activity decreases. Brain activity becomes more predictable. Brain injury, loss of consciousness. Brain activity becomes less entropic, more predictable. Deep sleep, same thing, more predictable and consciousness lost. Go into REM sleep. Now there's dreaming activity. The entropy of activity, you can see this bar chart, jumps up to near waking levels. So this was the intriguing possibility. What happens when you throw a psychedelic at this model and you find that the ordinarily very high entropic level of ongoing activity increases further. And of course, so does the richness of content of subjective experience. So for me, it's a neat principle. Of course, now the quest is to break it, what's its limit, but it's a useful principle, I think, going forward. It's well replicated. Our original work was with Hirschartner, it was with LSD, psilocybin and ketamine data, all showing the same thing. Now we've looked at DMT, it's a similar profile. We've actually looked at different environmental conditions when looking at the entropy or complexity of ongoing brain activity, found that the differential between placebo and drug, in this case LSD, is greater when people have their eyes closed, when there's less going on in the external environment. Introduce a bit of music, that differential shrinks. Actually, the absolute entropy goes up a little bit because it does. You've got more information content with music playing. You open your eyes, all of a sudden flooding the brain with photic information. Entropy goes up. It does on placebo because there's more informational richness. It goes up a little bit with LSD, absolutely. And then movie watching, even more information above the fixation cross of just opening your eyes, and entropy is maximal LSD with the movie watching. But the differential is less. And that's perhaps the most important principle here. The biggest difference in entropy, the biggest drive in difference is with the drug and with the least going on in the environmental conditions. This is the last model, I think, that I'm going to introduce to you. I'm looking at the time now. I've got my stopwatch going. I wanted to stop on 40 minutes. We've got about 6 minutes maybe to go. And so I'll end on this Rebus model. At least phonetically it works. Relax beliefs under psychedelics. And it rests on hierarchical predictive coding. Really, that's the inspiration. So let's get into the nitty gritty. And one way to demonstrate the principle is to to show it working in the mind. The fact that you can read scripts like this are, according to the Bayesian brain principle, because of our top-down priors driving our experience of the world, creating shortcuts, if you want, in processing. We see, I usually say movies like this, but that joke's getting a bit tired. Even with the joke, people, I think, I'm looking at it now and I still see that hallucinated motion. And that's according to the Bayesian brain model, because of our top-down priors. You see something like this in the world, typically there would be motion. You would have a revolution of the ball into this sort of, space. There is no motion. It's a static image, but our priors are driving that hallucinated percept. So the model, the Rebus model says, we could think of all sorts of beliefs or assumptions. Perceptual ones are just useful because they're kind of, tangible. But the ones that are arguably more relevant in psychiatry are more abstract, negative assumptions about oneself, about life in general, And so under the psychedelic, the model that I'm proposing is that, yes, it works at perceptual level, but also all the way up to the most abstract, cognitive, affective levels, where the weighting of the top-down aspect of functioning is lightened. There is a de-weighting, a lightening, a relaxation of whatever is encoding the top-down in this system. So, on that principle, we looked at traveling waves. I saw some excellent work by a French team, Van Rulen and his colleagues, showing a relationship between traveling waves recorded with EEG and predictive coding. And so I thought, wow, this is a great example to throw the Rebus model at. And Chris Timmerman, give them our DMT data. Let's do a collaboration, which we did. And the principle is this: when you close your eyes, the flow of information ordinarily is top down. It's from the frontal cortex to the back of the brain, the occipital cortex, probably because the brain is saying nothing to see here. You have your eyes closed, the flow is top down. You open your eyes, different situation, all this photic information flooding in now, and the direction of the traveling waves switches. It's bottom up. It's from the occipital cortex up to the frontal cortex. So we asked the question, what happens with DMT? Inject in DMT, very rapid onset. People feel the effects, maybe even within a minute. Peak effects around about 2 minutes, very intense. And we see something very similar to what you see with photic stimulation, just opening your eyes. We see a very dramatic switch, a flip in the direction of the traveling waves. People have their eyes closed, we're looking at them, that's confirmed, but the lights come on and they're seeing things. They're immersed in this DMT world. They might see other beings. It's a world analog. It's like dreaming. You're immersed in this other world. And the brain is sort of recapitulating that if we're looking at traveling waves. So this, in a sense, is sort of, intriguing potential support for the Rebus model. We've also looked at the principal gradient. I talked about that earlier, running a PCA on the connectivity profile of the whole of the brain. What axis or gradient explains most of the variance, the principal component, principal axis. Well, it's this one here that separates the transmodal from the unimodal cortical regions. Throw in a psychedelic and there's this, I like to call it an entropic effect. It's like a gas expanding in the space. It's now homogeneous. It spreads out. There's a collapse in the principal gradient. When that was first published on comprehensively, Margulies et al., it was explicitly linked to hierarchical organization. So you could say this is evidence for a collapse in the functional organization of global brain function under here DMT. Here DMT. We've submitted on LSD. We've also shown it with psilocybin. So again, it seems to be robust. Okay, I think I've got about 30 seconds or something like that. we finished our trial. It's a 2-arm trial, double-blind, RCT, psilocybin versus escitalopram for major depressive disorder, not treatment-resistant depression. 59 patients, either 2 sessions of psilocybin, 25 milligrams, quite a high dose, followed by paralleled by 6 weeks of placebo capsule ingestion, 43 of those, only 2 dosing sessions with psilocybin, 43 daily capsule ingestions. The other arm, you get just 1 milligram of psilocybin, presumed to be inactive, so placebo dose, but we standardize expectation. Everyone will receive psilocybin, so people are prepared, all the preparation is the same, clinician and patient. But in that condition, after the first dosing session, the very next day, it's 6 weeks of escitalopram, going up to 20 milligrams, solid dose of escitalopram for the final 3 weeks. So 43 of those. And the first draft has been circulated with the other authors, with Compass Pathways, who provided the drug for the trial. Revisions being done at the moment. Results are remarkable. Internally embargoed, I would say, before we submit. Hopefully submission isn't too far away, maybe weeks. Really exciting, multi-dimensional separation. I'm so tempted to say how, but lots of different, not just the sort of classic depressive symptoms, but you look at general functioning, things that matter to people, sexual functioning, and separation. There are also side effects, tolerability, favoring a particular condition. So really exciting, and can't wait to release that to the world when it's been properly reviewed. And I'll end it there. Thanks to the team. Very much a team effort, all of this work. And we have the freedom to do investigator-led research because of the philanthropy support that's come in for our center. So eternally grateful to have that freedom, and I think it allows us to do more exciting science rather than watering it down for public funders or company interests or whatever. So I'll end it there, and thank you very much for listening.
So thanks, Robin, for yet another amazing talk. You have this ability to knit together stuff from neuroscience and imaging and clinical psychiatry in a way that's just incredible. We have a couple of questions in Q&A that I'd like to transmit to you, but also if anybody who's listening, if you have questions for Dr. Carhart-Harris, you can just type them into the Q&A box. So the first question is from Ottavio Vitolo. And he asks, do the thalamus and basal ganglia play a role in the modulation of the transmodal cortex by psychedelics? Some subcortical influences.
Well, there is an alternative model, perhaps to the Rebus model. I think it's maybe complementary rather than competing. And it's from Franz Vollenweider. I think Mark Geyer was on the sort of original conception of this thalamocortical model, thalamocortical gating, reduction in gating, so more information flowing, I think, through the thalamus was the model, or up from the thalamus to the cortex, more in that sort of, if you want, bottom-up direction. Thalamus is part of the limbic system, I think you could say. And so in that sense, it's consistent, and there's a bit of dynamic causal modeling evidence to support that direction of information flow. And so in that sense, yes, I actually think Matthias Lichty found something implicating the thalamus. We've looked as well. I think there might have been a slight increase in thalamus to cortical connectivity. I can go back and look at that. It might be in a paper in Schizophrenia Bulletin. I think we looked at that, where we looked at the default mode network and dorsal attention network and other potentially inversely correlated or at least orthogonal networks to some extent with the default mode. And we looked at the thalamus there. as a kind of control, and a modest, I think it's a modest increase. So yes, the basal ganglia, I can't say anything specific. I think we saw drops in blood flow in our first psilocybin study, but that was always a bit of a tricky one to interpret because of potential complications around vasoconstriction. I'd need to revisit the data, I think, on that one. Yeah.
Cool. So there's another question, and I think they say antipsychotics. I think they mean psychedelics. How heterogeneous is the response to psychedelics? And are there people who don't respond well? If so, do you have any clue as to why? I think that's actually a really important question.
That's a critical question and quite difficult to address in the limited sample sizes that we have so far. But it's certainly something that's right up there on the priority list for questions of real importance to address. One way we've done this is to collect naturalistic data, because there you have a bigger spread. I think what happens in these clinical studies, whether we're fully aware of it or not, is that we can easily recruit a somewhat homogeneous population. And we're probably recruiting people that seem to have kind of, classic kind of almost reactive depressions, maybe where there's a sense that, it might be these cases might be somehow less complex. I think particularly that might have happened in the more recent MDD trial we did. And so because of that, you're sort of diluting out some of the potential interesting variants where you would have the complex cases that they don't respond well, or there is even maybe an iatrogenic response. Why that is, I don't think we know enough to answer that at this stage, but from the bigger data that we have from our naturalistic work, this is online surveying, it's not just retrospective, it's prospective. So we track people from ahead of taking a psychedelic and then track them across time. We've got a couple of thousand, I think, data points on that now. And we see things that they're really speaking to basic principles of set and setting, that if you trust the people that you're with, you're taking the psychedelic with therapeutic intention. There's music, actually, and you like the music. That was a key factor. If people take the psychedelic in a group, a sense of group rapport, something called communitas, seems to predict positive outcomes. In terms of personality, I don't think we found it yet. I mean, there's a sense that there's something in the complexity of the relationship, the therapeutic alliance, that's important, but quite what the right measure is, I don't think we found it yet. There's a measure of trait absorption, our ability to be immersed in stimuli, whether externally sourced or internally, relates to general sensitivity to a psychedelic, and there seems to be a relationship there with a particular Serotonin2A receptor genotype as well. But in terms of what is it in someone's character or state that predicts a less positive outcome from a psychedelic, I don't think we're there yet. I know I'm going on a bit, but there's one further thing to add. We recently did a sub-analysis in adolescents, kind of for obvious reasons, seeing the way things are taking off, what's happening with the multi-site trials, and then thinking, whether the scientists do it or the regulators ask for it, sooner or later, people are going to be asking, what about adolescents? Very good reason to be thinking about that population. And if the intervention could be prophylactic, then, even more so. And so there we found, well, we did see positive outcomes, but we saw that the usual mediators of positive outcomes related to the acute experience, the so-called mystical type experience or peak experience, and ego dissolution specifically, it didn't predict positive outcomes in the same way as it did in adults. It wasn't a flip of the relationship. It was just, the usual strong positive relationship. The bigger the ego dissolution, the better you do as an adult. Wasn't true in adolescence. So, maybe that speaks to special care in the psychological support, or just, special care more generally if going into such a population. We know, of course, adolescence and psychosis risk. So, yeah.
I think we have time for one more question. I'm going to take Rick Doblin's question. Rick actually gave Grand Rounds a few months ago in May for us, and he just wanted to know a little more information. Protocol design for the new Ibogaine study, subjects. You can answer that in 3 minutes.
Well, it's great, Rick. Right. Finger on the pulse as always. That study's developing as we speak. So the truth is, we don't yet have a crystallized protocol, but there is, some resources to do that. And what we've always done in mine and David Nutt's team is to do things iteratively. Small steps first. Ibogaine has a particular safety risk profile, and so really we want to ask, start by addressing the question, how does it work in the brain? No one knows that. It has this rich pharmacology. What does it do to brain function? Is it comparable to the classic psychedelics? And so we want to go in healthies, carefully screen for cardiac functioning and cardiac monitoring during. So play it really careful, dosage, dose finding, healthy volunteers, and do it that way. So it's going to be a brain imaging study, and that's the focus to begin with. If we can get it through as well.
Yeah. So for those of you that are unfamiliar with Ibogaine, it's shown promise, particularly in potentially treating opioid use disorder. And there are sort of Ibogaine clinics where people use that. The major medical risk with Ibogaine that separates it from other psychedelics are cardiotoxicity. So that's what Robin was referring to. I think we're at time. Robin, one more time, thank you so much. Great talk. Appreciate you being here.
I appreciate the invitation. I enjoyed it. Thank you.
Take care.